Introducing the ArundiMax medium, a scientifically formulated peptone-enriched culture medium designed for the in vitro mass propagation of Arundina graminifolia, also known as the bamboo orchid. This innovative culture medium was the result of collaborative effort between Nur Fatin Amira and Nurin Edina from the Faculty of Science of University Malaya and Nur Analiza Watima, Dr. Fazlin Haslinda and Dr. Siti Syairah from the Faculty of Pharmacy from UiTM Puncak Alam. Their backgrounds in plant biotechnology, bioinformatics, and pharmacognosy contributed to shaping the direction and success of this formulation. The development of ArundiMax was guided by three key objectives. Firstly, to enhance in vitro germination, shoot formation, and plantlet quality of Arundina graminifolia. Secondly, is to create a standardized, user-friendly Arundina graminifolia culture medium preparation system. And lastly, to support sustainable propagation for Arundina graminifolia conservation and commercial purposes. By enriching the medium with peptone, an organic nitrogen source rich in amino acids and vitamins, we aim to unlock the full potential of bamboo orchid micropropagation. Arundina graminifolia, commonly known as the bamboo orchid, is a tropical species native to Southeast Asia, including parts of China and India. It's easily recognized by its bamboo-like stems and delicate pink lavender flowers, which bloom throughout the year. While this vibrant color is the most common, a rare white-flowered variety also exists, adding to the orchid's uniqueness. Beyond its beauty, Arundina graminifolia is valued for its medicinal properties, traditionally used for its anti-inflammatory and antimicrobial benefits, making it highly sought after in both landscaping and herbal medicine. Despite its beauty and value, Arundina graminifolia faces serious challenges when it comes to propagation. Traditional methods, such as seed sowing or division, often result in very low germination rates, which is lower than 1%, and poor shoot development. Even under ideal conditions, the seeds struggle to sprout, and the few that do tend to grow slowly with limited regeneration potential. So, why are orchids difficult to grow naturally from seed? First, orchid seedlings face intense competition from other plants for sunlight, water, and space. At the same time, they're vulnerable to predators like insects and fungi, making survival rates incredibly low. Second, orchid seeds are extremely small and contain no stored nutrients. To germinate, they depend entirely on a partnership with specific types of mycorrhizal fungi. Without this symbiotic relationship, the seeds simply cannot grow. Finally, while orchid seeds are easily dispersed by wind, they are so light that they often land in places where conditions are not right. With no nutrients and no fungi, most fail to develop. But the threat to this species goes far beyond cultivation. In the wild, Arundina graminifolia is increasingly under pressure. Habitat destruction, driven by deforestation for agriculture, urban development, and infrastructure, continues to shrink its natural environment. At the same time, illegal collection for ornamental trade removes wild plants before they can reproduce. And climate change further disrupts the delicate balance of temperature, humidity, and rainfall that orchids need to survive. These combined threats are causing a steady decline in wild populations, making effective propagation not just a scientific challenge, but an urgent conservation priority. To overcome these propagation challenges, plant tissue culture offers a reliable and efficient solution. By growing orchids in a controlled, sterile environment, we can bypass the limitations of natural germination and accelerate plant development in a large scale in a shorter amount of time. With this in mind, we developed ArundiMax medium, a specialized culture medium enriched with pharmaceutical-grade peptone. This provides readily available amino acids, organic nitrogen, and vitamins that support rapid shoot formation, the development of protocorm-like bodies, and robust biomass growth. ArundiMax is designed to give Arundina graminifolia the optimal start it needs, from seed to strong, healthy plantlets. The name ArundiMax reflects both the plant it was designed for and its performance-driven formulation. Arundi is derived from Arundina graminifolia, the target species for propagation, while Max represents the medium's goal to maximize growth, efficiency, and propagation success. Together, ArundiMax stands for a specialized solution tailored to unlock the full potential of this remarkable orchid.
Here is the detailed composition of the ArundiMax medium. It includes a balanced mix of macronutrients like ammonium nitrate, potassium nitrate, and calcium chloride, as well as micronutrients such as zinc sulfate, boric acid, and copper sulfate. It is also rich with vitamins like thiamine, nicotinic acid, and pyridoxine, along with glycine and peptone for enhanced nitrogen supply. Importantly, an abiotic elicitor is added to stimulate the plant defense responses and boost growth potential. The medium is supplied in 4 easy-to-use sachets, each clearly labeled for precision and convenience. To prepare, sachets 1, 2, and 3 are mixed into distilled water, bringing the total volume to 1 liter. The pH is then adjusted to between 5.3 and 5.5 using a pH meter. Once balanced, sachet 4 is added and the mixture is stirred thoroughly until fully dissolved. The solution is then sterilized by autoclaving at 120 psi for 20 minutes, ensuring a clean, contamination-free environment for micropropagation. After cooling, the medium is ready to be dispensed into culture jars. Our findings further validate the performance of ArundiMax. In Figure 1, you can observe the clear visual differences in growth between the control and the base media. And in Figure 2, quantitative data shows that ArundiMax had the highest success rate in PLB proliferation and number of shoots generated. Seeds cultured on ArundiMax achieved a germination success rate of 90% compared to only 30% in the control group. Shoot production was also enhanced, with ArundiMax generating 270 shoots while the control produced just 90 shoots. These results speak to the medium's effectiveness in delivering faster and more consistent growth. ArundiMax medium offers advantages that go beyond the lab, delivering impact across social, environmental, and economic dimensions. On a social level, ArundiMax supports orchid conservation and serves as a valuable tool in education and research. Its user-friendly format makes plant tissue culture more accessible to students and institutions, encouraging knowledge sharing and increasing public awareness of biodiversity preservation. Environmentally, ArundiMax provides a sustainable alternative to wild orchid collection, helping to protect natural populations of Arundina graminifolia. The healthy plantlets it produces can support reforestation and habitat restoration efforts. And with fewer field cultures, the process is both resource efficient and environmentally responsible. Economically, the medium is cost effective. With its high germination and shoot formation rates, it reduces time, resources, and labor. This makes it ideal for mass production in nurseries, commercial horticulture, and orchid farms. The four-sachet system minimizes preparation errors and streamlines workflow, offering reliability at scale. And beyond horticulture, Arundina's medicinal properties, such as anti-inflammatory and antimicrobial, mean that ArundiMax supports not just plants, but future breakthroughs in herbal medicine and pharmaceutical research. Arundina graminifolia deserves a future, and ArundiMax medium makes that possible. With proven effectiveness and user-friendly design, it's a reliable solution for orchid conservation, research, and commercial growth. ArundiMax medium, where innovation meets propagation.